Hello all, welcome to the presentation Base Excision Repair Mechanism. Base Excision Repair Mechanism is a DNA repair mechanism wherein the altered bases of the DNA is removed or replaced. In base excision repair, glycosylase, an enzyme which recognizes and removes the damaged base by hydrolysing the glycosidic bond. The abasic sugar is then removed from the backbone by endonucleolytic action. Endonucleolytic cleavage also removes apurinic or apyrimidinic sugars that arise by spontaneous hydrolysis. After the removal of damaged nucleotide, DNA polymerase and DNA ligase act upon the damaged strand to restore. Let's see what is the role of glycosylases in base excision repair mechanism. Normally, cells will have different DNA glycosylases with different specificities. For example, uracil glycosylase, which specifically removes damaged uracil. Like that, there are 8 different glycosylases present in the cell. So this is one of the example of fail-safe glycosylase and its action on base repair excision mechanism. So here, due to oxidation, the guanine became oxygen, and the fail-safe glycosylase comes into action and restores its function. So it corrects the damaged base. How does glycosylase detect damaged base? Let's see that. Glycosylases diffuse through the minor groove of the DNA until a specific kind of lesion is detected. Glycosylases remove the damaged base by a mechanism called base flipping. So what is base flipping? The damaged base project out or flip out of the DNA double helix so that glycosylase can go there and bind to that particular flipped out base and can be removed out. So this is how glycosylase detect the damaged base. So the damaged base basically flips out of the DNA helix so that glycosylase goes there and removes it. One other important thing is that the glycosylase will remove only the damaged base. It will not cleave the DNA backbone. It will only cleave the particular base, not the backbone. So the DNA backbone remains intact. Only the damaged base is removed. So here is the overview of how the base excision repair mechanism works. Here you can see, so you know that in DNA normally adenine, thymine, guanine and cytosine are present. Uracil is not normally present in DNA. It is present only in the mRNAs. So in case if uracil is incorporated into the DNA, so it will create problem during the replication. So this need to be removed. If there is an uracil present in the DNA, that need to be removed from the DNA helix. So the enzyme uracil glycosylase will come into action here, which will specifically remove an uracil, but it will not cleave the DNA backbone. Only the damaged or the wrong incorporated base is removed. So uracil glycosylase come here and cleaves that particular uracil and removed off from there. So which will create, what will happen when a base is removed is that there will be a gap present in that particular site. It is called the abasic sugar. The sugar which without a base is called the abasic sugar. So now the DNA has to, the cell has to replace that particular base with the correct one. So the DNA polymerase is the enzyme which is responsible for synthesizing the nucleotides or incorporating the nucleotides. But the problem here is that DNA polymerase cannot act on if there is cannot act on this particular site because there is no nick or there is no free OH group for the polymerase to act upon. So this particular site is called the AP site. It is called called the AP site. Here the AP endonuclease or exonuclease will come into action, and it will cleave or nick the DNA backbone so that the DNA polymerase can act upon that particular site and fills in or it can incorporate the base which is removed. Instead of uracil, it will fill what is complementary to the other strand. So this is the overview of base excision repair. Here you can see that there is a DNA, the DNA with a wrong nucleotide incorporated. Let's see, I have given an example as uracil. So in DNA, uracil is incorporated in this case, so that need to be replaced. So uracil glycosylases come here into action, and it removes only the uracil without nicking or without cleaving the DNA backbone. So that particular site is called the AP site, apurinic site or apyrimidine site, where the AP endonuclease will come into picture, and it will cleave or create a nick there. So now you can see here a nick is created. So now with a free 3-hydroxyl group, you can see here AP endonuclease cleaves here and makes a 3' OH4 polymerase 1 to act upon. So once the nick is created, DNA polymerase will come here. Actually, DNA polymerase has two activities. One is the exonuclease activity, and the other is polymerase activity. So due to the exonuclease activity, it will chop off certain bases from this nick region, and at the same time, this because of the polymerase activity, it fills in or incorporates what is complementary to the bottom strand or the antisense strand, let's say. And once it is done, DNA ligase will come and seal the nick, and the DNA is repaired. Now you can see from here there was a uracil here. Now that has been removed and replaced with the correct base. This is how base excision repair works. Hope you understood it. If you like the video, please subscribe. Thank you.